Class 11th Biology Chapter 2 Biological Classification. Now we start. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis of classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed, and that include all plant and animal respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organism, and photosynthetic, green algae for example, and non-photosynthetic, for example fungi, organism. Classification of organism into plant and animal was easily done and was easy to understand, but a large number of organisms did not fall into either categories. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organism have hence undergo several changes over the time. Through plant and animal kingdom have been constant under all different systems, and the understanding of what groups organisms be include under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. R.H. Whittaker in 1969 proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Animalia, and Plantae. The main criteria of classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Gives a comparative account of different characteristics of the five kingdoms. This table 2.1, this is characters, five kingdoms, Monera, Protista, Fungi, Plantae, Animalia. Cell type, cell wall, nuclear membrane, body organization. Mode of nutrition. In Monera, cell type, prokaryotic, cell wall, non-cellulosic, polysaccharide plus amino acid, nuclear membrane absent, body organization cellular, mode of nutrition autotrophic, chemosynthetic and photosynthetic and heterotrophic, saprophytic and parasitic. सारे ही तरीके के हैं इसमें. प्रोटिस्टा, cell type eukaryotic, cell wall, some present in some, nuclear membrane presents, body organization, cellular, mode of nutrition, autotrophic, photosynthetic and heterotrophics. Fungi, cell type, eukaryotic, cell wall, present with chitin, nuclear membrane present, body organization, multicellular, loose tissue. Mode of nutrition, heterotrophic, saprophytic, sometimes parasitic. Plantae, cell type, eukaryotic, cell wall, present, cellulose. प्लांटी में सेल वॉल सेलुलोज की बनी होती है, फंजाई में काइटिन की होती है कुछ में, और प्लांटे में न्यूक्लियर मेम्ब्रेन प्रेजेंट, प्लांटे में बॉडी ऑर्गेनाइजेशन टिश्यू लेवल और ऑर्गन लेवल, प्लांटे में मोड ऑफ न्यूट्रिशन ऑटोट्रॉफिक फोटोसिंथेटिक. एनिमेलिया, सेल टाइप eukaryotic, cell wall absent, nuclear membrane present, body organization tissue organ system. Mode of nutrition heterotrophic, holozoic or saprophytic. The three-domain system has also been proposed that divided the three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain and thereby a sixth kingdom classification. You will learn about the system in detail. Let us look at five kingdom classification to understand the issue and consideration that influenced the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm and angiosperm under plant. The character that unified this whole kingdom was that all organisms had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, for example cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular ones. Says, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic groups, fungi, the autotrophic green plant, though they also showed a characteristic difference in their wall composition. The fungi has chitin in their walls while the green plant had cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, earlier placed in algae within plant and both having cell wall, with Paramecium and Amoeba, which were earlier placed in the animal kingdom, which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. With this happened because the criteria for classification changed. This kind of changes will take place in future. to depending on improvement in our understanding of characteristics and evolutionary relationship over time. An attempt has been made to evolve classification system which reflect not only the morphological, physiological and reproductive similarities but it also phylogenetic that is based on evolutionary relationship. In this chapter, we will study characteristics of kingdom Monera, Protista and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, we will dealt separately in chapter 3 and 4. Kingdom Monera. Bacteria are the sole membrane of the kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitat such as hot spring, desert, snow, and deep oceans where few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape. The spiral coccus, plural cocci, the rod-shaped bacillus, plural bacilli, the comma-shaped vibrium, plural vibro, the spiral spirillum, plural spirilla, cocci, round-shaped bacteria, bacillus, rod-shaped bacteria, spirillum, it's a connected flagella-like bacteria, and vibro is a comma-shaped bacteria. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group shows the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty area, halophiles, hot spring, thermoacidophiles, and marshy area, methanogens. Archaebacteria differ from other bacteria in having different cell wall structure and this feature responsible for their survival. In extreme conditions, methanogens are present in the gut of several ruminants animals such as cow and buffaloes and they are responsible for the production of methane biogas from the dung of these animals. Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as a blue-green algae, have chlorophyll A similar to green plant and are photosynthetic autotroph. In figure 2.2 you can see this. This is a nostoc, a blue-green algae with a filamentous. This cell is a heterocyst. This is a mucilaginous sheath over the cells. The cyanobacteria are unicellular, colonial or filamentous. Freshwater, marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheet. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. For example, nostoc and anabena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrate, nitrite, and ammonia and use the release energy for their ATP production. They plays a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria. They are most abundant in nature. The majority are important decomposers. Many of them have significant impact on human affairs. They are helpful in making curd from milk, production of antibody, mixing of nitrogen in legumes, fruits, etc. Some are pathogenesis causing damage to human being crops, farms, animals, and pets. Chlorella, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fusion, as you can see in figure 2.3, dividing bacteria. This is a bacteria is dividing in two of the daughter cells, and this is two different DNAs of the bacteria, and this is a cell wall, is inside a cell membrane. This is a cytoplasm. Sometime under unfavorable conditions they produce spores. They also produce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to other. The mycoplasma are the organism that completely lack of cell wall. They are smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic in animal and plant. Kingdom Protista. All single-celled eukaryotic are placed under Protista, but the boundaries of this kingdom are not well defined. What may be photosynthetic protistans to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellate, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the others dealing with plant, animals, and fungi. Being eukaryotes, the Protistans' cell body contains well-defined nucleus and other membrane-bounded organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote in formation. Chrysophytes. This group includes diatoms, golden algae, decimates. They are found in freshwater as well as marine environment. They are microscopic and float passively in water current. Planktons. Most of them are photosynthetic in diatoms. The cell wall forms two thin overlapping shells which fit together as a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposit in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oil and syrup. Diatoms are chief producers of the ocean. Dinoflagellate. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigment present in their cell. The cell wall has a stiff cellulose plate on their outer surface. Most of them have two flagella, one lies longitudinally and the other transversely, in furrow between the wall plate. Very often red dinoflagellate, example goniolix, undergo such rapid multiplication that they make a sea appear red. Red tide toxins released by such large number may even kill other marine animals such as fishes. This is datum euglenoid. Majority of them are freshwater organisms found in strength water. Instead of cell wall, they have protein-rich layer called pellicle, which make their body flexible. They have two flagella, a short one and long one. Through they are photosynthetic in the presence of sunlight. When deprived of sunlight, they behave like heterotrophs by predating to other smaller organisms. Interestingly, the pigments of euglenoid are identical to those present in higher plants. For example, euglena. This is euglenoid. You can see the two flagella, one is longer and one is shorter. Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaving engulfing organic material under suitable conditions. They form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spore at their tip. The spore possess true walls. They are extremely resistant and survive for many years, even under adverse conditions. The spores are dispersed by air current. This is slime molds. As you can see, this is a cell wall and these are the spores, a black dot. Now, protozoans. All protozoans are heterotrophs and live as predator or parasite. They are believed to be primitive relative of animal. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, seawater or marsh. They move and capture their prey by putting out pseudopodia, false feet as in amoeba. Marine forms have silica shell on their surface. Some of them, such as entamoeba, as parasite. Flagellated protozoans. The members of this group are either free living or parasitic. They have flagella. The parasitic forms cause disease such as sleeping sickness, for example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of presence of the thousands of cilia. They have a cavity called glut that opens to the outside of the cell surface. The coordinated movement of the rows of cilia cause the water laden with food to be instilled into the glut. For example, paramecium. Sporozoans. These include diverse organisms that have an infectious spores-like stage in their life cycle. The most notorious in Plasmodium malarial parasite, which cause malaria, a disease which has a strengthening effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread or rotten fruit. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves and are due to parasitic fungus. Some unicellular fungi, for example yeast, are used to make bread and beer. Other fungi cause disease in plant and animal. Wheat rust causing Pakinia is an important example. Some are source of antibiotics, for example penicillium. Fungi are cosmopolitans and occur in air, water, soil, and on animal and plant. They refer to grow in warm and humid place. What you ever wondered why we keep food in refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infection. With the exception of yeast, which are unicellular fungi, are filamentous. Their body consists of long, cylinder thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tube filled with multinucleated cytoplasm. These are called coniocyte hyphae. Others have septae or cross wall in their hyphae. The cell wall of fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate and hence called saprophytes. Those that depend on living plant and animal called parasites. They can also live as symbionts in association with algae as lichens and with root of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spore formation called conidia or sporangiosperm or zoospore, and sexual reproduction is by oospore, ascospore, and basidiospore, or various spores are produced in distinct structure called fruiting body. The sexual cycle involves the following three steps. First, fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Second, fusion of two nuclei called karyogamy. Third, meiosis in zygote resulting in haploid spore. When a fungus reproduces sexually, two haploid hyphae or compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cell 2N. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage N plus N, that is, two nuclei per cell, occurs. Such a condition is called dikaryon, and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi from fruiting body in which reproduction division occurs, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation, and fruiting body from the basis of the division and of the kingdom into various classes. Phycomycetes. Members of Phycomycetes are found in aquatic habitat and on dividing woods in moist and damp place or as obligate parasite on plant. The mycelium is aseptate and coniocytic. Asexual reproduction takes place by zoospore, which is motile, or by aplanospore, which is non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar, anisogamous or oogamous. Some common examples are mucor, which you can see in image 2.5a. This is mucor. Rhizopus, a bread mold mentioned earlier, and albugo, the parasitic fungi on mustard. Ascomycetes, commonly known as sac fungi, the ascomycetes are mostly multicellular, for example penicillium, or rarely unicellular, for example yeast. Saccharomyces, they are saprophytic decomposers, parasitic or coprohylas, growing on dung. Mycelium is branched and septate. The sexual spore are conidia produced exogenously on special mycelium called conidiospore. Conidia on germination produce mycelium. Sexual spores are called ascospore, which are produced endogenously in sac like asci, singular ascus. These asci are arranged in different types of fruiting body called ascocarps. Some examples are Aspergillus, claviceps, and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edibles and are considered delicious. Basidiomycetes. Commonly known from basidiomycetes are mushrooms, bracts fungi, or puffballs. They grow in soil, on logs and trees stumps, and in living plant bodies are parasitic. As parasitic, example, rust and smut. The mycelium is branched and septate. The sexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains of genotype. The resultant structure is dikaryon, which ultimately give rise to basidium. Karyogamy and meiosis take place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in fruiting body called basidiocarp. Some common members are agaricus, mushroom, Astrago smut, Puccinia rust fungi. Deuteromycetes, commonly known as imperfect fungi because only a sexual or vegetative phase of these fungi are known. When the sexual form of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that an asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage other and placed under another class. Later, when the linkage were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stage of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes produced only by asexual spore known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, which are large number of them. They are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Coleotrichum, and Trichoderma. Kingdom Plantae. Kingdom Plantae include all eukaryotic chlorophyll-containing organisms commonly called plant. A few members are partially heterotrophic such as insectivorous plant or parasites. Bladderwort and Venus flytrap are examples of insectivorous plant and Cuscuta is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae include algae, bryophyte, pteridophyte, gymnosperm and angiosperm. Life cycle of plant has two distinct phases, the diploid sporophytic phase and the haploid gametophytic phase, that alternate with each other. The length of haploid and diploid phase and whether these phases are free living or dependent on other vary among different groups in plant. This phenomena is called alternation of generation. You will study further detail in this kingdom in chapter 3. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell lack cell wall. They directly or indirectly depend on plant for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have definite shape and size. Higher form shows elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Silent features of various phyla are described in chapter 4. Virus, viroids, pyron and lichens. In 5 kingdom classification of Whitaker, there is no mention of lichens and some acellular organisms like virus, viroids and pyrons. These are briefly introduced here. All of us who suffered the ill effect of common cold or flu know what effect virus can have on us. Even if we do not associate it with our condition, virus did not find a place in classification since they are not considered true living. If we understand living as those organisms that have cell structure, the virus are non-cellular organisms. They are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, take over a machinery of the host cell to replicate themselves, killing the host. Would you call virus living or non-living? Virus means venom or poisonous fluid. Dmitry Ivanovsky in 1892 recognized certain microbes can casual organism of the mosaic disease of tobacco. These are found to be smaller than bacteria because they pass through bacteria-proof filter. M.W. Jering in 1898 demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plant and named the new pathogens virus and called the fluid as Contagium vivum fluidum, infectious living fluid. M.W. Stanley in 1935 showed that viruses could be crystallized and crystal consist largely of protein. They are inert outside their specific host cell. Viruses are obligate parasite. In addition of protein, virus also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA and viruses that infect animals have either single or double-strand RNA or double-strand DNA. Bacterial virus or bacteriophages. Viruses that infect the bacteria are usually double-stranded DNA virus. The protein coat called capsid, made of a small subunit called capsomer, protect the nucleic acid. These capsomers are arranged in helical or polyhedral geometric form. Virus cause disease like mumps, smallpox, herpes, and influenza. AIDS in human also caused by virus. In plant, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunt growth. Viroids in 1971. Teodinier discovered a new infectious agent that was smaller than virus and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in virus, hence the name viroid. The RNA of viroid was of low molecular weight, pyrons. In modern medicine, certain infection neurological diseases were found to be transmitted by an agent consisting of abnormal folded protein. The agent was similar in size to virus. These agents were called pyrons. The most notable diseases caused by protein by pyrons are bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and its analogous variant Korsakoff disease, CJD, in humans. Lichens. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycobiont and the fungal component is mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral, nutrition and water for its partner. So close is their association that if we once saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in polluted areas.